Hi, Dhruv. How are you now? I hope you're doing fine now. Okay, so I've shared the live stream link with you all. So if any one of you misses out on the meeting, you can join there. So while we are waiting for others to join, let me just take you quickly and tell you how you need to sign up yourself and create an account and get the access to the material on the portal. It's really good to see so many new names today. It's really good that you all. Okay, sorry for the echo. I'm sharing my screen and so who all of you have already signed up on Learn? Let me know by text on the chat, maybe by sending out one or yes. And those who are not signed up, I would request them to please sign up on Learn. I'm sharing the link with you all in the chat. Here is the link. While others are joining, it would be great if you sign up so that you can go along with us in the session itself. And those who are already signed up, I request them to please update their profiles. So all you have to do is please go to your profile by clicking on your name. And I would want you to add a profile picture and also link in your social accounts so that first of all your name, it should be the full name that you would want on your certificate, because this is the name that would be reflecting on the certificates as well. And why I'm asking to link your social, it's just because we keep on featuring our best profiles, so we would want to have your social links with us as well. And I would also explain more advantages of having an updated profile in the meantime. I think many of them have joined. Let's wait for 2 more minutes. Hi, Kakshil. Hi, Ikiba. Ali, can I know in which country are you placed so that I am aware of your time zone? Hi, Ikiba. Okay, so I think let's get started. We have our participants and others can join in the meantime. So I welcome you all to today's research fellowship meeting. So before I start with the meeting, let me first of all introduce you to the research fellowship program, as we have a lot many new participants here as well. So this Bioinformatics Research Fellowship Program is a structured program that guides students through various areas of big data bioinformatics research using practical examples. And during this program, we would go through several high quality research publications and would learn about applications of computational biology in projects through these publications. This kind of experience would allow beginners to try computational biology techniques on public domain data, making it possible to work with large files and extract meaningful information from patient samples, animal models, cell lines, and microbiota as well. The program would provide support and mentorship, And therefore it is an intensive research program that would be involving training tasks, access to our online sessions, and a guide on the implementation of learned skills to proposed research ideas as well. So let me quickly introduce ourselves before we begin with the session. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. OmicsLogic is an international program that is running in 5 different regions with over 10,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. My name is Sonalika Ray, and I'm the OmicsLogic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of a bioinformatics community managed by Pine Biotech. And I'm honored to have been working with a team that collectively has an amazing experience in the area, including the other co-founders of the company, Dr. Alfred Tauber, who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation. And one of his major projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Then we have Dr. Leonid Brodsky. He's the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics, and over the course of his career, he has developed multiple novel algorithmic approaches to biomedical data analysis. These have been widely used in basic and translational research in biotechnology, pharmaceutical R&D, agriculture, and basic research as well. Our platform is a result of hard work by some of the people who are mentioned here on the right. Julia Panov is an expert bioinformatician and has helped prepare many of the materials that we would use. Avi Titovsky is a computer scientist and leads the development of T-Bioinfo platform that we would use. Dr. Harpreet has an expertise in the application of statistical machine learning techniques and biomedical data and development of prediction tools and databases as well. And she looks after the curriculum development, bioinformatics, and data science course development for the company. Then we have Dr. Mohit Mazumdar with us, who is an expert bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. And over the past several years, Dr. Mohit Mazumdar has been developing collaborations with universities as a mentor and project lead for OmicsLogic training and research fellowship. So let's start by signing up to learn.omicslogic. This is an educational portal that would help you get started on your bioinformatics research journey. So those who joined us just a while ago, I was telling that we need to sign up on the OmicsLogic portal. I would share the link again, and those who have signed up, it's good to have them here. You can now update your profile as I've told you. So I would be waiting for you. You all can sign up now. I've shared the link. Let me show you how you can sign up. So here is the sign up page. You need to enter your mail ID. You would get a link, and then you have to create your password as well and then sign in. You can also sign up via your social accounts, and it would be an easy way to sign up on the portal, and then you would be good to go for your courses and other opportunities as well. So let's wait for the participants who have joined to sign up. Let me know once you are done. I would be waiting for response in the chat box. It would be great if you could tell me if you have signed up as a 1 or maybe yes in the chat box. Thank you, Rizwana. Thanks, Abhilash. Good, Suraj. Okay. So getting the access while signing up would let you go ahead with us in the session. Thanks, Poonam. Good, great. Okay, so now I would want to pass it on to the CEO of Pine Biotech, Ilya Brodsky. Over to you, Ilya.
All right, thank you very much, Sonalika, and welcome everyone to our session today. I want to go quickly over several aspects of this program so that we can hear from our research fellows that are joining and also those that are working on their projects. So as we move along, I want to introduce you to some of the key areas that this program is designed to address. One of the important aspects of the research fellowship program is to enable research. And so in a lot of ways, what we try to do is we try to provide tools and an environment where you can effectively analyze big data, access our network of mentors, and also go through some of the training that we provide as asynchronous online courses and live training, which are programs. So in the next few minutes, I'll introduce you to some of these things so that everyone knows how to benefit from these tools and opportunities. So OmicsLogic program resources combine training materials, data analysis tools that are GUI-based and allow you to process big data effectively, training in coding, and introduction to research topics using different types of omics data. These were developed as a part of the TBioInfo platform. And once you register on the learn.omicslogic.com portal, you essentially get access to both of these portals and platforms. On the portal itself, you will find that the key area is to complete your profile. And the reason for that is because all of the information is going to be stored there. So you can see that you can update your profile picture, social media. You will start tracking your achievements and activity. And you will find all of the information about the program right here down on the bottom. So this is the research fellowship program. So if you don't have a badge and you've already joined, that means that you have not been added to the program. Once this profile is complete, you can always navigate to the different programs that we have and find other opportunities for you to continue and explore different areas. For example, right now we have a program that's running on bioinformatics for infectious diseases that includes specific courses, access to Zoom interactive sessions like today, and provides you with a sense of community because we offer a Slack channel where you can communicate directly with the mentors, as well as those that would like to participate in a one-on-one session. You can request a session with your mentor. Now, those of you that still don't have that badge, please reach out to marketing@pine.bio, and Sonalika, who introduced the program, will add you to the program. Once you're inside the program, you will go to your profile tab and find there the different programs that you're enrolled in. For example, as you can see, Introduction to Bioinformatics, Transcriptomics, or Bioinformatics for Infectious Diseases, and you can track your progress in that program by completing individual sessions. Importantly, we have a set of points, which is a system that we designed so that we can track your progress and give you a sense of where you are related to where you should be to be able to start an independent project. So I'll talk about that in a little bit. For those of you that are a part of the research fellowship, you can reach out to us. Many of the sessions are going to be available to you for free, and some of them will require payment, but you can reach out to us about that. So another thing that I wanted to ask all of you to do is to expand our network. We have a group on social media, on LinkedIn and on Facebook. So if you, I will paste the links. If you are a part of the research fellowship already, we invite you to join these social media groups. And if you are not a part of them yet, then you can also mention that you are going through this fellowship. This allows us to see in our network those of you that are members and those of you that are mentors and connect you in the future. So the other thing I mentioned is that we have a basic training resource where you can use Introduction to Bioinformatics coursework on genomics, transcriptomics, metagenomics, epigenomics, and data science. All you need to do is go to the OmicsLogic portal and find a course that is interesting to you or that matches your level. So what you can do is, if you see under your profile again, you should have a basic subscription, or it should say starter. If you are a part of the research fellowship already, it should say starter. So let me show you what that looks like. Again, this is important to check because you're going to be seeing different coursework that is premium. And so all of you have access to the free courses. Once you sign up, the free option is just sample a lot of the coursework. But those of you that have either paid for a research fellowship already, or you were granted a scholarship through the Louisiana Biomedical Research Network, you will get a starter subscription, which gets you into courses that are premium. So those that have a star next to them, you will find those. I will just give you an example. So you will find the courses with a star. Those are accessible only for those that have a starter subscription. So make sure that you have that accessible. If you don't, reach out to marketing at Pine Bio and we will help set that up for you. Now, another important opportunity that the OmicsLogic portal and the community provides is access to these specific webinars that are free for all. So if you have joined our community, you can gain access to all of the free webinars, and we have regularly maybe 2 or 3 webinars a week that you can join and benefit from some introduction to a topic or an expert that speaks about a variety of topics. So these include training programs, and it also includes the Psiomics Club. It is a club that is operated by one of our research fellows, Ruth Vaja, and she is going to be sending invitations to all of you that are interested about how to register for that club. Those of you that are research fellows can also join those sessions to talk about your completed projects. Now, importantly, on the portal, we don't have just courses, we also offer project examples. So the project examples are essentially papers that have been digested and translated into tutorials that you can follow, performing the same analytical steps and getting to the same outputs as described in the paper. And so this is a way to get started with your research topic. You can find a paper that is most relevant to either the data type that you have or a topic that you're interested in to learn about that and try to perform similar analysis. A lot of that is going to be done on the T-Bioinfo platform. So once you have signed up on the Learn OmicsLogic portal, you will get a demo account on the Big Data Analysis platform. So because all of you are here, I want to just quickly try this. If you already created a profile, let's navigate to server. .tbio.info, and please try to log in. And if you can log in with your account, you will see your email address right here. So if you're able to do that, please let me know in the chat so I know that you can log in, because we're going to be using this throughout the research fellowship. And so if you're just getting started today, by creating an account, you should be able to access this portal as well. Thank you, Faria. Okay, especially for those of you that are joining today for the first time, this is why we want to check this today. If you do not have access to this, then you need to reach out to us and let us know, and we will look into it. Excellent. Okay, thank you everyone for sharing that. So let me briefly talk about why the research fellowship and what is essentially the objective of this research fellowship. So I'm excited to, okay, Ituraya, if you have an invalid email and password, please share that with Sonalika by email and maybe share a screenshot. Okay, so the research fellowship is an opportunity to access all of these opportunities together, get the training that you need, and start working on an independent project. So here are some examples of previous participants that have gone on to publish, have gone on to present their research at different types of poster sessions, and also even lead as a mentor themselves, training others from what they have learned. And so as you think about how others have gotten to that stage, let's break it down so that we have a step-by-step sense of how you could go about choosing a project, developing that idea into something that you can execute in your free time, and then present your findings in front of your peers. The first step is obviously to select a topic. So when you think about a research project, one of the critical things is to start off with an impactful topic that you would be interested in and that others would be interested in, but also something that actually could be done with data that is available. So as we go through this process, I will guide you through some practical steps that you can take to identify the specific topic. Then think about the audience, presenting and asking them whether this is something that they would be interested in, and then finding a data set that you can actually work on during this research fellowship program. A lot of times, getting started means following experts. And so what we offer for you to get started and get a sense of how things work, and this is where I believe all of you should get started with, are some practical examples on a variety of topics that are available already on the learnomicslogic.com portal. These include projects that cover a variety of data types, a variety of analytical methods, and a variety of applications of bioinformatics to solve problems in biomedical research, in the clinic, as well as biotechnology and even agriculture. And so as you look through some of the projects that we have already available, this should give you some ideas about what you can do in your own time. Now, importantly, you want to get to a stage where you take what you've learned through your training and start developing your own independent project. And so that process starts from literature review. A lot of times you can see a particular general topic and then go through making that topic a lot more specific by going through literature review. So in literature, you will identify gaps and answer questions, controversy, and debate to identify that specific focus for you to work on your independent project. Ultimately, that helps you define your question and hypothesis. And what you will do throughout the meetings that we have as a part of the research fellowship is go through this process of presenting what you have learned about, then talking about your general topic and the background, as well as what you have found in literature, and then defining that research question or hypothesis. Next, you will have the time to evaluate different data sources and data types that we will introduce you, such as the National Center for Biotechnology Information and other repositories that are more specific, like Malaria Gen or NCBI virus. And there you will identify a project data set that you will work on during your research fellowship. So then the research fellowship really is broken down into 3 specific steps. The first one is introduction, where you get to learn and get introduced to the topics. Evaluation, where you get to develop your research proposal and find the reference publications and data sets. And then as you go through the development of the project itself, you essentially apply and practice what you have learned, but on a project of your own. Now, very soon, we're going to be launching this as an interactive process on the portal itself that starts from what we started with today. You will update your profile with a good profile picture and a completed profile where you can update your information about you and connect to social media. Then you will have a goal, and that practical goal will get you focused on achieving the type of expertise that you need to work on an independent project. And then we will offer you a course that actually guides you through the steps on how to make a project that is impactful and how to apply rigorous research using computational biology tools to a particular data set. So then once you are done, you will have a chance to actually put together that project on your profile, and it will be added so that anyone can see what you have been working on, what is the data set that you've used, what are the tools and the methods that you have done, and see your whole project presentation right there on your profile, including a video presentation and the description of all of the methods. So we will guide you through steps like creating a profile on GitHub, posting there the curated dataset description that you have found for the dataset you've used. We will talk about how to describe the methodology that you used in your research. And eventually you will also add there a set of scripts that you've used and others can see and maybe use themselves. So this is a little overview of what that will look like in the end. And to understand how that all fits in with the process of this research fellowship, I want to mention some general steps that we will go through, which include comprehension and analysis, where you have to explore the data and really understand what the data could be used for. Investigation, where you get to practice your critical thinking and asking questions, translating them into technical set of criteria that you can apply to get rigorous research results, and then practice writing, editing, and preparing the project for submission. And an important part is to be able to present this both to your mentor and to the community, which we have joining this session today. So ultimately, you will go through these 10 defined steps that we think will guide you to an excellent outcome. And we have some experience getting others to get to that point. So step 1, you will identify a topic by conducting literature review. Step 2, you will identify a data set, reference publications, and define a project question or focus of your research. Step 3, you will make a table of metadata for your data set to include group names, number of samples, and type of data in each data set. On step 4, you will conduct exploratory analysis such as PCA, summary statistics, or general visualization, and share the results with your mentor by email or on Slack to get feedback and discuss analysis steps. Step 5, you will conduct data analysis, which will be in-depth, and prepare all of the results in a presentation. Then you will review biological databases and literature, comparing your results with other findings and identifying what part of your findings are actually novel, and then address any issues that are highlighted. On step 7, you will present to your mentor, peers, and experts for final review. On step 8, you will finalize the presentation, record it, and submit the final project for the program. And then you will get help from our mentors to identify a poster session, an opportunity to publish or present for your mentor the project that you have worked. And then you will finish by submitting the final project on your profile. For Rhea, yes, I will explain literature review. In fact, we have a whole course on literature review that we will be publishing soon. So that's a great question. The place to start is to do a search on NCBI for PubMed and finding some review articles that you can go through and think about what is the topic and what are some of the questions that people are asking about that topic. But we will turn to that later. Does anyone else have any questions about these steps? Okay, if you have any questions, you can put them in the chat and we'll make sure to address them. So finally, the objective, your first objective, is to summarize in a structured way your project as a poster. So you will talk about what is the topic and what is the objective. Then you'll describe the methodology, results, and conclusions or discussion at the end. Now, this is something that will guarantee a positive outcome for all of you. Some of you are very dedicated and have a lot of free time, and your objective is maybe to publish. For others, you will see an opportunity to get trained and use your skills in a meaningful way. And some of you will want to work with others. So you don't have to necessarily work on everything on your own. Sometimes it's very helpful to work with others. So if you are in this community, that's why we also have the Slack community. It's very important that you're able to interact with others, convince others that you have a great project to work on, and then form that collaboration. Now, bioinformatics is a very collaborative kind of a discipline. Everybody has to work with others. So you actually have to get to know the other people, understand how to work together and maybe divide up the work, and then apply it in a meaningful way. So we ourselves are oftentimes a part of a project, and we can support your project in a meaningful way by addressing some of the gaps that you might have in your research. So this is an example. We had a collaboration with the University of Ghana, where a whole group was dealing with genetic diversity of malaria in the country of Ghana among different areas in that region. And some of our team has actually contributed to doing the analysis or writing or even presenting the final product. So sometimes this will be your own independent project where you've done so completely on your own. And we have a lot of examples of people that have done just that. And others actually work with other team members and are able to collaborate so that they can present. Now, after you complete your project, we also will offer you an opportunity to get out there and be more visible in front of others. And so here are some examples of featured profiles that we've done for previous participants. We highlight their achievements in submitting their preprints on bioRxiv. We highlight their achievements when they actually publish. And we have a number of people that are publishing, including undergraduate students and even high school students. And we even work with people that are already in research. And so they're able to utilize the training that they get to really make more from bioinformatics components in their research. And that includes people that are in different universities as PhD students, master's students, but also high school students as well. So all of these examples, I hope that we're showing you today as examples of those that have done similar things like what you want to do today. And as you go through the process of the fellowship, these are some of the critical guidelines that we provide you with so that you can be more effective. Now, let me briefly introduce you. We do have, beyond the research fellowship meetings that are going to be like this today, we have a whole set of summer research mentors that could offer you help and guidance in making your project more successful and helping you get to a research result that is more impactful for you, depending on what you want to do with these project ideas that you have right now. And so you already probably met Dr. Harpreet Kaur. She has a PhD in bioinformatics. She's an international author on many publications, a reviewer, and an expert in cancer biology. So if anyone has those kinds of projects, I think she's the best person to help you. Now, she's going to be helping most of you in any case. So if you have any technical questions or if you want some guidance, she would be the first person to turn to. Besides Dr. Harpreet Kaur, we have a number of other mentors that we will be able to introduce you to once you have a good presentation to share. So once your presentation has the exploratory data analysis, the topic, your plan for analysis, maybe even some preliminary results, that's when we can potentially match you with a mentor based on where you are, what are your objectives, and whether they have the expertise that you need. And we have Dr. Sona Vasudevan, who is professor and a program director and expert in systems medicine, but very passionate about teaching and helping other students get to where they want to be. Clinton Kunha, who has a double master's degree both in molecular medicine and in bioinformatics. Dr. Giuliano Chavez, he is a PhD in bioinformatics and expert in neurodegenerative diseases as well as glioblastoma. Sahil Sethi, who is a PhD student in bioinformatics, but also has a master's in health informatics and is an expert on big data in COVID and health disparities, as well as Dr. Laura Harris, who is a program director and a PhD in bioinformatics and an expert in microbiology. So we do have this great community of mentors. This community of mentors continues to grow. But to be able to work with these people, you have to first complete your initial project presentation, and you will see some of those examples today. Now, ultimately, what we hope will happen during the summer is that most of you will get to a place where your project is presentable. And that's why August 21 to 22, we're going to have our Omics Research Symposium. In this research symposium, we're going to be talking about the cutting edge research and applications of bioinformatics, and we'll have some guest speakers to speak about that. We'll talk about different opportunities for undergraduate, master's, and PhD programs in bioinformatics. We will have their recruiters and who will speak about industry internships and job opportunities for those of you that are looking to develop a career in bioinformatics. We will talk about effective resources for training and education for those of you that are involved at different universities and those of you that are a part of student communities that could benefit from that. And we will have prizes for best high school, college, and citizen science research projects. So that's an opportunity to earn some money, expand your network, and highlight what you have done over the summer. So again, I welcome all of you to join our social media groups and stay in touch. Also, continue to monitor our emails. We send a lot of emails from Sonalika, who will be sharing opportunities for training and for different opportunities like this one. And we also have a YouTube channel that highlights some of those sessions that are going to be helpful for you. Okay, so I will just address quickly Victoria's question. She's saying, what if I want to learn in multiple fields which I have deficiency, like metagenomics, chemoinformatics, methylation analysis? If I'm restricted to one project, am I going to have the chance to learn all the fields I'm hoping to learn? So that's why I mentioned that we have these asynchronous courses. You can go through those independently. We have training programs. You can always join the first few sessions to learn about that program. And if that's something you're interested in, you can reach out to us and you will have access to some of that. Or you can join the full program with certification for a separate registration fee. And you have the opportunity to work in the research fellowship for 3 months or 6 months, which gives you enough time to work on one project and get it done. And then potentially, if you want to work on other projects, you can join those as well. So it's really up to you. Okay, so now I want to pass it on to Dr. Mohit Mazumdar and Dr. Harpreet Kaur to guide us for the rest of the session and explain how all of you that are joining for the first time can start off by introducing yourselves. Thank you.
Thank you, Ilya. Hello, everyone. So my name is Mohit, and I've been working on bioinformatics projects for a while now. So I did my PhD in computational biology, and during that time, I've been able to work on several grants that were addressing some of the key objectives. Like I got to work on grants from Department of Science and Technology, Department of Energy, and also grants from NIH, and also some of them were from Japan and Indo-Shastri. So all of those international, objectives that were defined on those grants were to lead to some translational work, which was one of the objectives for me to do. So, second. Yeah, thank you. So, so during that time, I learned a lot, and then I signed up for a company to work on bioinformatics projects for pharma. And during that time, so let me just share my screen just a second. So during that time, I got to learn about how pharma is utilizing these resources and bioinformatics resources, and that actually is influencing a lot of their drug discovery pipelines, and that is actually helping them to shorten the whole drug discovery timeline and also save a lot of money. So they were into it, and a lot of data scientists being involved in pharmaceutical research that also like goes into the clinical trials, data management, all of that. So here I've been working with students and universities and research groups. So with all that experience, my objective is to help you all come up with project ideas. Whosoever is coming up with project ideas, to develop that project idea into a project, real project, as well as for those who are coming up as without a background of bioinformatics. So to start from the very basic and understand where to start from, how to begin, where to find information, where to find data, which courses to take, all of that. So in this during this time, I've been also working with, as I said, universities to develop courses. So universities and faculties who wants to implement these courses and teach them to their students, and all of that. So we have all these programs running in different places. So today I just wanted to tell you a little bit about what I did also during my PhD was related to machine learning. And here we are working with several types of data sets that are available publicly. And one of the biggest advantage in today's world is that there's so much of information which is available openly. And that is also due to the fact that many of these publications requires an open access to data, and that makes us, all applicable to do some new research on these data sets that have not been explored fully because they contain so much of information, and there are so many connections that different types of data sets have. And so exploring those and coming up with projects and working on those projects to come up with some of the solutions like patient stratification in the sense of clinical research, and then how you can use that information for disease classification diagnostics, and then response to therapies, which is precision medicine. So one of these things that we have been working on, and we are also creating lessons and courses so that you can go through this entire process and understand that how, I mean, from a data set, how you can come up with all these, uh, discovery terms like biomarker discovery, toxicity, finding the toxicity, drug target identification, or molecular mechanism of action, drug repurposing and repositioning. All of these scientific research that you see that many good labs are publishing, you are also capable of doing it by exploring the data and utilizing some of the tools that we have put together for you. So there are several types of projects that you think of when you are thinking about bioinformatics, and that is so exciting at the same time because you get an opportunity to work on several aspects, right? You work on sequences, you work on special characteristics of those sequences features, like different research data has different sort of features. Some are age dependent, some are time dependent, some are location dependent. So all of those things makes it like a variety of things to look at. And that actually, this multidisciplinary field gives that holistic understanding from coming from different types of data is actually maybe something that is an ultimate goal of ours, to understand everything, right? System biology, how the system works. So that integration of data and something that we are working with, and obviously biomarker discovery for pharma projects, and we have had examples on glioblastoma. We will have a session on the psiomics that Ilya said, which will be about also biomarker discovery. So then we have these opportunities from finding solutions. So first we have to identify the problem. So we started with basic understanding of what it is, what the technology is, how to apply the technology by learning through examples, and then you come up with your own research project and you find the problem. Okay, this is the problem. This is the protein. I mean, this is the gene. Now what to do with it? I mean, how to make it not do what it is doing incorrectly. So in that sense, where the strategy comes in, their cheminformatics comes in, their molecules comes in. So that's what one of the aspects of what pharma works on, and how machine learning is applied into this. So these are some of the applications. And similarly, I mean, in the context of how bioinformatics can be applied, and as I was telling you that it's very exciting because there's so many aspects. Like you can look at the networks, like how they are communicating amongst each other. So you can draw networks amongst, I mean, even within protein, you can draw a network for the atom interaction and how they are interacting, and how the structural changes are making that differ from one state to another. Similarly, network analysis can be done for sequence at a sequence level to understand disease patterns and biological systems. And similarly, like all of these things are exciting fields and lets you to discover new things, and those new things are available for you to discover. But yeah, obviously you would need to go through a way. I mean, what we do is that we follow a lot of publications and we try to translate them into something that is more understandable for everyone, for students, and to be able to come up with their own research. So I would also like to invite you for this upcoming free webinar, which is on June 23rd. Sorry, not June 23rd. This is wrong. So June 25th it is. It is on June 25th. Let me correct that. Yeah. So on Friday we have the webinar, and I'd like to invite you for the session also. And we have mentors, as Ilya have already shown you that. So as I was telling you, like one of the outcomes of these sort of going through this research process is to be able to come up with research experience, and maybe like if you go through this entire process and find results that are really interesting, you can come up with something like a publication also in a very short period of time. That is possible. So this is what we have experienced so far, working together with many smart and very hardworking students who want to do really well to apply this and come up with these nice research projects. So I would now like to maybe invite Dr. Harpreet, so that we can also hear from some of the participants who are working and right now, working on these projects and going through this entire experience. And I mean, you can always write to me and feel free to ask any questions that you have. I will look into the chat. So now I'll just invite Dr. Harpreet to. Okay, thank you, Dr. Harpreet.
Thank you, Dr. Mohit. So, Sonalika, can you just make me co-host?
Yes, ma'am.
Thank you. So, hello everyone. My name is Harpreet Kaur and I have a PhD in bioinformatics. And I have done my master's in molecular biology and biochemistry. And I'm always interested in analyzing those molecular mechanisms in human disease or different human disease conditions, especially the cancer. So since, as we know, there is a continuously enormous NGS data being generated from the patient, so I believe one can analyze such data to understand biological and molecular processes that is taking place in underlying specific disease conditions. So I joined my PhD at CSIR Institute of Microbial Technology, and I have done my PhD in the field of liver cancer. So my PhD research was on biomarker discovery for the diagnosis and the prognosis of the liver cancer using bioinformatics. So with the Pine Biotech, currently I am curriculum developer and research consultant, and I am mentoring number of the research fellows. So I'm looking forward to work with you. Now I think we should start with the presentation. Maybe Sonalika, we can proceed with the presentation from the research fellows now. Yeah.
Yes, ma'am. Thank you so much, Dr. Harpreet. Before we start with the presentations, we have a few new research fellows with us. I would want them quickly to introduce themselves so we know about them. So can I have Dana Rusan? Can you please introduce yourself to us? Hi, Dana. You can unmute yourself. Okay, so if Dana is not available, maybe Abhilash, can you please? Yes, yes,
I
yes,
can
Dana, go ahead.
introduce myself.
Yes, thank you.
Okay. Now I'm studying a master degree in microbiology, and my thesis will be on viruses and studying NGS data, and using nanobore as a technique so to study a whole genome sequencing of this virus. And actually, I'm interested in studying bioinformatics related for drug discovery and for infectious disease.
Okay, thank you so much, Dana. It was good to know about you and your research interest. Abhilash, can you please introduce yourself?
Hello, my name is Abhilash. I'm a PhD student at South Dakota School of Mines, and I'm currently working on metagenomics and transcriptomics analysis of.
Sorry to interrupt here. It would be great if when you're introducing yourselves, and Sonalika, maybe you can do this as well, turn on your video. It would be nice to kind of have a face behind the name.
Yeah.
Thank you.
Yeah. Hi, my name is Abhilash. I am a PhD student at South Dakota School of Mines, and currently I am working on metagenomics and metatranscriptomics analysis of sulfate-reducing biofilms, basically the biofilms which corrode metals. Yeah, and in this research fellowship program, I am interested in learning more about coding to analyze metadata. As of currently, I use Galaxy online platform, but I want to use R and Python to analyze metadata. That's why I have joined this program. Thank you.
Thank you so much, Abhilash. Hi, Iman. Can you please introduce yourself next?
Hi, I'm Opini. Hi. I am Iman Tourih. I'm assistant professor of medical genetics, working now in Tulane University School of Medicine and Department of Surgery. I was graduated from medical school, had master and doctorate degrees in medical genetics, and then I had a diploma degree in bioinformatics, but it was very basic, and I didn't go in depth into genomics, transcriptomics, and big data analysis. My research interest is cancer bioinformatics. But actually, I started to be fond of bioinformatics since 2014, when there was no available sources in my home country. So I started self-learning and attending workshops and learn a bit R coding for visualization. And to capture as much as I can, I applied what I learned in peer-reviewed publication. That was useful. And that's great job from your side, that all what we are going to be learning is to be in publications and posters and something valuable as well. And therefore, I felt there is a need for machine learning as well. I started to know more about Python coding. I'm not perfect in any of these, what I mentioned now, but I am trying and I want to learn more. As a geneticist, there are available public repositories in big data, and that needs to be going more in depth to unravel, to find new discoveries from these hidden genomic and transcriptomic signature. So I joined here the course in order to have a more comprehensive view, more analysis, to share, to expand my network with other bioinformatics and share experience with them, and to achieve more and to enjoy diving and exploration together with my peers. I want to discover more about drug discovery, metagenomics, methylation analysis, and GWAS analysis. I want to learn all the tools that I come across with the bioinformatics. So I'm hoping during the 6-month course of this fellowship that I upgrade myself in this field and teach others as well in my campus in my home country. Thank you again, guys.
Excellent. Thank you very much. Nice to have you here.
Thank you.
Thank you, Iman. Looking forward to your work. Faria, can I have you next, please? Yes, you're audible.
Hello, ma'am.
Hello. Yes, Faria, you're audible.
Yes, I'm Faria Brar. I'm a resident of Jeddah, Saudi Arabia. But I'm currently doing BSc Biotechnology from Delhi Jamia Hamdard University. And I'm just a student right now. So I have participated in research fellowship program to explore more on cancer or Alzheimer disease.
Thank you, Faria. It is nice to know about your interest. Poonam Sen, can we have you next, please? Poonam?
Hello,
Yes,
am I
yes.
audible?
Yes, you're audible. Thank
Yeah.
you.
Okay. Okay. Yeah. So my name is Poonam Sen, and I have done masters in plant science and masters in international relations. And I'm from India. Right now, I'm working as a botany faculty here in India. So I have joined. I've also like recently joined the Pine Biotech Research Fellowship, and I don't have much exposure to computational biology, so I'm very much excited to learn from all of you. My research interests are basically plant molecular biology and stress response, and I also want to explore about the epigenetic regulations, and yeah, so that's my intro. Yeah.
Thank you so much, Poonam.
Welcome.
Thank you, thank you, thank you. Rucha, are you ready with your presentation?
Yeah, I am.
Yes, so I've given you the access to please share your screen. Over to you, Rucha. You can start with your presentation.
Okay.
Rucha, if you could also please start with a brief introduction about yourself so others can get to know you. And if you don't have the option, turn on your video, that would be great.
Yeah, sure. I'll do that. But the thing is, my camera, there's a problem with it. So please, just this time.
No problem.
So, okay. Yeah, so I'll be sharing my screen now. Is my screen visible?
Yes, it is.
Okay. Okay, so hello everyone. My name is Rucha Konetti and I'm from Bangalore, India. So I'm currently studying biotechnology at Ramaya Institute of Technology. So I've previously done molecular docking studies on drug-resistant tuberculosis and SARS-CoV-2. But I've always been interested in how we can use genomics or transcriptomics and in the analysis of various diseases. So that's why I joined this research fellowship program, and I'm really glad I got a chance to participate. So today I'll be sharing with you what project I chose to do, and that's Alzheimer's disease. So Alzheimer's is a neurodegenerative disorder, and it affects individuals over 60 years of age or in people with trisomy 21. And commonly, the symptoms include dementia and other non-cognitive symptoms like delusion or agitation, etc. And neuropathologically, and this is the most important thing, there are formation of beta amyloid aggregates and the tau protein deposition. This is why the disease is caused mostly because of these deposition. So, uh, so that is why I chose my data set as from this bio project. This project may consider non-demented control brains and AD brains containing only the amyloid beta plaques or both the plaques and the tau pathology. So currently, what happens when amyloid beta and tau pathology increases in the brain is unknown. So that is why this project was probably done. And so this project identified two types of microglia, that is AD1 and AD2 associated with AD. And so AD1 was associated with the amyloid beta plaques, and AD2 microglia were abundant with the tau pathology brains. So identifying these microglia might help us, like, tell us more about how exactly AD is caused and why. So based on this, I chose to do my project. And so here there are two types of samples, oligodendrite and neuronal cell samples, taken from two regions of the brain, occipital cortex and the occipitotemporal cortex, or the fusiform gyrus. And these types of cells were identified by their biomarkers, that is the OLIC2 transcriptional factor and the neuronal biomarker respectively. So from in both these cell samples, there are three types of groups, that is AD, CTR and CTR plus. CTR is basically the control samples. CTR samples have no amyloid plaques or tau protein deposition as seen here, and CR plus have the amyloid plaques but no tau deposition. So the difference between this and AD is that although they have the amyloid deposition, these brains are not demented. But in AD, there is a distinct dementia that was observed. So, and of course, in AD, there is both amyloid and tau deposition. So, coming to my data set, there were 15 samples totally, consisting of the oligodendrite and the neuronal samples. And in each, there were CTR, CTR plus, and AD as shown here. So, and coming to my research questions, what I want to ask was how the AD and control groups are different from each other in both the cell types. And later on, I would like to identify, like, gene expression in the OC and OTC regions of the brain in both the cell samples among the three groups. So first I did PCA among the cell samples, and this is what I got. The red dots clustered together show the neuronal cell samples, and the blue dots show the oligodendrocytes. And this PC1 versus PC2 indicate 62% of the total variance. And then I did PCA among the three different groups in both the cell samples. So on the left, you can see oligodendrocyte PCA, and on the right, you can see neuronal PCA. And so here, you can see in the oligodendrocytes that this is PC1 versus PC3, showing a total of 40% of the variance. And the red dots are clustered together here, that is the AD samples. The green dots are here, that is the control samples with no amyloid beta plaques. And the CTR with amyloid beta plaques are shown in the blue dots. Similarly, you can see for the neuronal samples. So I've done till here. I'd like to know what I can do further on. Thank you everyone for listening.
Thank you, Rucha, for sharing. And because first thing you need to understand based on this PCA plot, what are the different outliers? Can you, I mean, in the first part, if we look at, there are the two outliers, like the green one and this blue one, and that is control. Because to me, it is still not clear. It looks like CTR plus is actually maybe we can say it's like a stage where this pathology starts, or the beta plaques just start getting accumulated, something like that, right?
Yes, ma'am.
It may be, I mean, this is the control. I don't understand, like control, simple control must be the normal sample. So here looks like that. Maybe the control plus samples. It's like where you just start, beta plaque start getting accumulated. Okay, so maybe now what you can do. Okay, so the first thing you need to do, remove these two outliers. If look, it looks like maybe this green one, not this one. I mean, on the left side, left side.
Yes, ma'am.
Yeah,
Which one?
yeah, this
Here.
one, this
Okay.
and this, and one at the top, that is blue one, that is,
Okay.
yeah, it probably
Okay.
they looks like these are the outliers. And maybe first thing that you need to do for particularly if you're dealing with this data, that is oligodendrocytes, dendrite
Sites, yes,
cells.
ma'am.
Yes. So I think first you need to remove that and maybe need to run PCA again. And then
Okay.
you can find out what are the differentially expressed genes between. Maybe here you can ask two types of questions: control versus AD and CTR versus AD, CTR plus,
Okay.
sorry, CTR plus versus AD. And this is the first thing that you need to look at, like what are the significant genes, maybe what are the important genes that can actually differentiate these two groups. So we need to
Okay.
identify the transcriptional changes at the first stage.
Okay. So I have to do differential expression, ma'am.
Yeah, using DSEC,
Okay.
because
Okay.
I
Okay.
do remember in your data it is already you have read count values. So the next
Yes,
you
ma'am.
need to prepare your data set to run DSEC analysis. Maybe here
Okay.
you can actually compare multiple groups at the first, just to understand whether you actually have, some important significant genes that can distinguish these classes of the samples.
Okay.
And then let's see how what you are getting out of that. And now I just
Oh,
want Ilya must add his points here.
okay.
Yeah, I just wanted to ask again. So, Rucha, what is the specific question that you're trying to answer with this analysis?
I want to know how, like this data set considers two different brain regions. So I want to see how the genes are expressed in those regions among the different groups here.
But why?
Because that's where the plaques and the top protein deposition occurs most often. That is AD
So,
neuropathology.
right. So I think that's the point that I'm trying to make is you need to think about what is the specific question. So if you go back to, I think, your second slide. So what happened was you introduced yourself and then you told us, yeah, maybe where's, okay. So this is a background about Alzheimer's disease.
Yeah.
And now you have a specific question. So as you explore this data set, and hopefully you've done some literature review, link
Yes.
your analysis to some important question that is still unanswered about Alzheimer's disease. So think about, why would you do all of this analysis? How will the results of the analysis help you answer that question? Once you get a set of differentially expressed genes, for example, why are they important and what will you find? What will be the potential impact of what you're doing with this?
Okay. Okay.
Okay.
Right. I'll do that.
And you can see someone is writing in the chat, Itoria. So we have three groups and you can make pairwise analysis and find the intersecting genes. So before you even go down to the steps of, what other people are suggesting, you always have to think of what is your objective? Why are you doing what you're doing? And where
Okay.
will that lead you to? After that, I think in the next session, just a couple more observations. We want to know a little bit more about why you selected Alzheimer's disease as your topic of interest. And when you present this, present it as if you are presenting to collaborators. So all of us here on the session, we want you to succeed and we want you to do something meaningful with your time. So how can we help you? Can we help you by making suggestions about bioinformatic analysis? Can we help you by focusing your attention on some important question that you could answer with your research? Could we maybe guide you to another project that somebody else is working on that maybe combining the two will make your project more impactful, right? We want to know
Great.
how can we help you as we're listening to this. And
Okay.
when you get to a point that you don't know what to do, it has to be connected to where you started. So you said, okay, so I did up until now I don't know what to do next, right? So
Right. Yeah.
you should from the beginning say, that's what you should announce. I have done my literature review, I have done my exploratory data analysis, and now I am at a place where I'm starting to identify the specific analysis question that I will focus on, right?
Okay.
And that's where you should get us to.
Okay. Okay, I'll do that. Yeah.
Okay, perfect, thanks.
Okay. Thank you very much.
Thank you so much, Ilya and Dr. Harpreet, for all these suggestions. Now, I would want to invite our research fellow Dhruv Mehra to please share his research project. Over to you, Dhruv.
Hello everyone. I hope I'm audible and visible.
Yes,
Hello.
Dhruv, you are.
Okay. Is my screen visible?
Yes, Dhruv, it is.
Okay.
Thank you.
So, hello everyone. My name is Dhruv Mehra, and today I'm here to present the progress of my research project called Transcriptomic Analysis of Esophageal Carcinoma. So, a quick introduction. My name is Dhruv Mehra, and I'm a graduate in medical biotechnology. So, I graduated last year from Amity University, Noida in India. And at present, I'm working as a research fellow at Pine Biotech. And talking about my past experiences, I've worked as an intern in research and standard department of a precision medicine company. And there I used to work along with my team on projects related to genomics and molecular diagnostics. And other than that, I've actually worked as an histopathology intern in a government hospital here in India only. And talking about my research interest, I'm interested in precision medicine, cancer biology, genomics, and transcriptomics. And through this fellowship, I'm expanding my horizons in the field of data science. So before starting, I'll just introduce my topic. So esophagus. Esophagus is actually a muscular organ that connects your pharynx to the stomach. And esophageal carcinoma is actually a malignant neoplasm of the esophagus. And the condition is actually associated with tobacco use, alcohol consumption, and deficiency of vitamin A, B3, and C. Along with that, there are several health conditions associated with the disease, such as esophagitis, celiac disease, and GERD. So talking about the epidemiology, so these figures are from the WHO Cancer Repository. These figures are from year 2020. And in that year, there were like 600,000 cases of esophageal cancers. And the mortality rate was somewhere around 540,000. And talking about the geographical distribution, Asian population has actually reported the maximum number of incidents. And these figures are actually quite surprising as it indicates the need for newer therapeutic, diagnostic, and prognostic approaches for the management of the disease. And that was the major motivation for me to, undertake this project. Before defining my hypothesis, I'll just explain my dataset. So for this project, I've actually used the TCGA esophageal carcinoma dataset. And the dataset actually consists of 198 samples, out of which 13 are normal samples. And there are 185 tumor samples. And like these are my preliminary hypothesis. So by this research project, I want to attain two major objectives. So first is to know more about the gene signatures involved in cancer progression. And second is to look for gene signatures or biomarkers associated with prognosis of the disease. And here is also an analysis plan which I've been following. So I started by preparing my data set. So I matched my metadata with the expression data. And then I took the average of the duplicate genes. And then I proceed with the PCA. And today I'm here to present the results of my DGE analysis. So this is actually a PCA from my for like when I was undertaking my objective one. So I actually divided my data set into as per the stages. So there was like normal, there was stage one, stage two, stage three, and stage four. And surprisingly, I actually got these two clusters, which were quite surprising for me. So there is cluster one on the right, and there is cluster one on the left. And then I looked into this cluster formation, and it turns out to be that these two are the histological subtypes of the esophageal carcinoma. So, in this PCA, like on your right is the esophageal squamous cell carcinoma, and on your left is the esophageal adenocarcinoma, and like the normal is diffused in between. So before like going, I'll just quickly describe these two subtypes. So esophageal squamous cell carcinoma. So it's a malignant epithelium tumor with squamous cell differentiation. So actually squamous cell cells actually form the innermost lining of the esophagus, also the epithelium. And in this cancer subtype, that epithelium, the squamous cells are actually affected, and they form the tumor. And like uh, in the esophagus, the middle third, like this esophageal squamous cell carcinoma is localized to the middle third of the esophagus and is associated with factors like tobacco use, alcohol abuse, and micronutrient deficiency. And likewise, we have esophageal adenocarcinoma, which is actually the malignant tumor of the esophagus with glandular tissue involvement. So this is somewhat different from the esophageal squamous cell carcinoma because it is associated with a condition called Barrett's esophagus. So Barrett's esophagus is actually a pre-neoplastic condition in which the squamous cells are actually converted into the glandular intestinal type of tissue, and which leads to the further esophageal adenocarcinoma. So here is a figure of the process of carcinogenesis. So first we have the esophageal squamous cell carcinoma. So this is the normal epithelium, and the like it is continuously insulted by alcohol, tobacco, and thermal injury, which leads to the subsequent phases of carcinogenesis. So like there is squamous hyperplasia, squamous dysplasia, and this which further results in full-blown esophageal squamous cell carcinoma. And likewise, we have the second subtype. So like the normal epithelium, then it's converted into the Barrett's esophagus because of the insult of the bile and the acid reflux, also leading to transversions, subsequently leading to the carcinogenesis, like there are conditions, dysplastic Barrett's esophagus, and then to the esophageal adenocarcinoma. There was like during my literature review, I actually found something really interesting about these cancer subtypes. So like in the graph A, uh, there like the esophageal adenocarcinoma is actually reported, and it was seen that majorly the developed nations have reported this cancer type, and it was opposite for the case of esophageal squamous cell carcinoma. All the developing nations have been reporting the cases of esophageal squamous cell carcinoma, and I guess that was the reason I couldn't even find a single paper which could actually, compare these two subtypes. And then I took my research forward and I plotted the PCA. So here is a PCA between the normal and the esophageal adenocarcinoma. So again, two clear clusters. So first and the second, again, there are outliers, and I did the same with the second subtype. So again, two clear cut clusters, first and the second. So likewise, I then proceeded with my differential gene expression analysis, and I actually performed this into three subdivisions. So in the first, I compared the normal and the esophageal adenocarcinoma, and then I compared the normal versus the esophageal squamous cell carcinoma. And then I actually compared the esophageal adenocarcinoma and the esophageal squamous cell carcinoma. So here is the pipeline construct. So for this differential gene expression analysis, I've actually used the DSEC2 uh, algorithm since I was supplied with the raw read counts. And here were the results for the differential gene expression analysis. So I actually found 8,745 genes which were differentially expressed between normal and esophageal adenocarcinoma with a pH-adjusted value of less than 0.05. Then, to see about the upregulation or the downregulation of the gene, I set up a log FC cutoff. So I found 1,473 genes which were upregulated, and there were 1,808 genes which were downregulated. Both like the log FC was greater than 1.5 in case of upregulated, and for the case of downregulated, it was less than 1.5 or minus 1.5. And there is actually a volcano plot describing the results. So volcano plot is actually a type of a scatter plot which which compares, which have like minus log of p-adjusted value on the y-axis and the log fold chain on the x-axis. So the dots in the red are actually differentially expressed genes, and we are actually interested in the red dots here, which are the upregulated and the downregulated genes. And likewise, I've actually compared the other group, like the normal versus the esophageal squamous cell carcinoma. And like I again found 10,000, more than 10,000 genes which were differentially expressed with p-value of greater than 0.05, more than 2,000 upregulated and downregulated genes. And then the final one, the esophageal adenocarcinoma and the esophageal squamous cell carcinoma. So surprisingly, there were 14,000 or more than 14,000 differentially expressed genes. And after setting up the log FC cutoff, I actually got 2006 genes, and which were upregulated, and 1612 genes which were downregulated. I actually tried to do a comparative analysis. So I've actually compared the case one with the case two. So here the case one is actually the differentially expressed genes in normal versus esophageal adenocarcinoma, and the second case is the differentially expressed genes with normal versus the esophageal squamous cell. And as a result of that, I've actually got common genes. So I've got 845 upregulated genes which are common in both the subtypes, and 1,013 genes which are downregulated in both the subtypes. And the reason why I did that was first because I wanted to know more about the common differentially expressed genes which were common in both the subtypes, like this region, which is, I hope the diagram is visible. So I focused on this common region, and the second reason being to look for subtype specific differentially expressed genes. So if I look at this area, so it will actually help in better patient stratification, and maybe I can get some insights about like various pathways involved in subtypes which are quite specific. And for the future, I'll be actually doing a pathway enrichment analysis, and I'm planning to conduct a more comprehensive literature review just to see, just for knowing the cancer subtype management. And maybe after that, I can get some good results, and I'll share them with you. Thank you.
Thank you so much, Dhruv, for such a wonderful presentation. Now, I would want Dr. Harpreet to please give the suggestions.
Okay, so thank you, Dhruv, for nice presentation. And I have a few things to say. The first thing, when you actually doing um, Venn diagram, so why you compared only two? Instead, you because you already have two subtypes, right? Instead of the two, I think you also
Yes, ma'am.
consider the third, where you are doing a differential gene
Both the
expression
subtypes.
analysis
Both the subtypes.
between ASC and ESSC. So that
Yes,
is
ma'am.
the another thing. But because as I know, this data was taken from TCG itself, right? So as you
Yes,
were
ma'am.
mentioning about your literature review, because some of the one subtype is more prevalent in developed countries, and another subtype is more prevalent in developing countries. So because in TCGA data, you also have ethnicity information.
Yes,
You
ma'am.
should know what are the Asians, or Americans or Latins. So I just want to see, actually, if you look at in your samples, whether you can also compare that part, because when you are talking about, like, there is a difference in even a subcontinents. So that is
Yes,
also
ma'am.
we need to understand whether there is actually ethnicity is also there. I mean,
Okay.
how these genes or these subtypes are associated with that ethnicity.
Okay, ma'am. I'll definitely look into that
Because
because, like,
yeah, in the
it
TCA,
is.
that information is provided.
Yes, ma'am. Definitely,
Race
because the
and
geographical
mhm,
distribution
yeah.
is quite different for both the subtypes. So I'll
Mhm.
definitely look into the ethnicity.
Yeah, I mean, the another information, there must be the race, which race they
Okay.
belongs to. So these factor you must consider, because when you are doing this analysis, and I thought that must be important. Finally, when you are
Okay, ma'am.
actually going to compare the things and going to interpret these results, maybe that would be interesting to understand that part.
Okay, ma'am. Definitely, I'll look into the this part and I'll share my results next week.
Okay, thank you.
Okay, I also wanted to mention a little bit. Um, so another type of a factor that is actually common with esophageal cancer is Epstein-Barr virus infection, and typically that's also reported, right?
Yes, sir. Yes.
So, the question about the cause of cancer is one question that you have.
Mhm.
The other
Yes.
question that you might have is what would be the treatment? Another question that you might have is early detection,
Yes, sir.
right?
Definitely.
So all of these factors are there, and you've done some exploratory analysis, and you just decided, I'm going to do now comparative analysis of gene expression. Once you do your gene expression, let's say you find some enriched pathways, or let's say you have some specific genes that you're interested in. Another question that you could ask is, how is the current treatment of those types of patients? And you have some treatment information in the TCGA data also, is
Yes.
changing for esophageal cancer, because it is changing. There are some common targets for cancer, and then there are specific targets for cancer. So I think
Yes.
that the place where you're at is a good place to pause for a second, consider the various factors that you have before going down to the comparative level. You already know what to expect, but before you know
Yes.
what to focus on, you have to maybe give us some idea of, out of all the factors, this is the actual difference that I'm interested in. Or maybe out of all of the potential metadata that I have access to, this is the type of metadata that I would like to focus on. And consider also other types of data. So you chose transcriptomic data.
Yes.
Why just look at gene expression? Why not also look at mutation data, for example, or methylation data?
Okay. So I got inspired by this one essay, like there is PAM50 essay for breast cancer, which actually describes a lot. Like there is like what would be the prognosis, best treatment. So I thought of something very similar for this, but definitely I'll look into these points and then I'll work on that.
Okay, and I do see some 5 points that again was in the chat, and I think that that's some good feedback, Iman. Okay, thank you, Iman. Sorry, I don't know from your username. So number one, why a jar was written in the workflow despite seeming not used? Do we need to multiple methodology when we choose? I think this is just a figure from the platform. You get thousands of genes, why not raise the fold change threshold? For that question, I'm not sure that you need to raise necessarily the fold change. I think what you need to do is downstream analysis, group them by pathway or gene ontology. Then she's mentioning maybe you reflect risk stratification in your objective. So if it is a risk stratification, you mentioned smoking, you mentioned other factors, there's viral infection as a factor. It would be nice to understand whether that is something that has already been explored. So have you read the publication that is associated with the whole data set?
Yes, sir. Actually, like the original TCGA publication actually talks about the data integration. So they have actually clubbed the transcriptomics and the mutational data. So they have actually clustered this cancer into subtypes. But surprisingly, like no one has actually compared these two subtypes. And like I was actually proceeding with that idea only.
Well, that's actually a good point. Maybe you could clarify that at the beginning. So you did review their publication and you know that there's a way to cluster patients, but no one really looked in depth at a particular cluster and how that difference. Also, yes, you did mention in your PCA analysis that these are groups or clusters. Maybe you could actually do that clustering and show us how those clusters separate. That's a good point. Okay, so then number 4, she's talking about in the objective, it was mentioned in intention to compare the 2 cancer types. Do you intend to do that with DSEC adenocarcinoma versus squamous cancer, or finding the non-intersectional gene list is enough? So I think here, you're doing this comparative analysis, but I think that at the moment, it's still a little bit exploratory because you haven't considered all the factors like Harpreet mentioned. Maybe after you've done that, you can show the results.
Okay, so I'll definitely look into the metadata and the raised points, and like I'll share my results next week. Okay.
Thank you.
Thank you so much, Dhruv, for a wonderful presentation. And thank you, Ilya, for the suggestions as well. Now, I would want to call our next research fellow, Jeevanjeet Kaur. She's from Amity University, Noida, and she wants few suggestions from Ilya. So over to you, Jeevanjeet. I'm making you the co-host. You can now share your screen. Over to you.
Thank you, ma'am. Hello, am I audible?
Yes, you're audible and we can see your screen as well.
Okay. Hello, everyone. I am Jeevanjeet Kaur from Amity University, and I'm a final year student of BTech Biotechnology, and I live in India. So I joined Pine Biotech for my summer internships 1 month back. So what I have completed is I completed the genomics course. For that, I have to complete the R code also. So I completed the R code one also. I done all the codes and done all the assignments and quizzes. Okay, sorry. Am I visible?
Yes, you are. Thank you.
Okay. And I had gone through the following papers, which are through my research interest. So what I basically learned is actually I had learned a lot of things, like everything is interconnected. It's not just the courses, but I had gone through many things. I had gone through the Wikipedia, gone through YouTube, Googled many things. So I learned the DNA structure and the basic concepts related with DNA, whether it's the structure, whether it's the histones, or whether it's like transcription and all. I had learned the R code, where I learned the loading of data, about the visualization of data through various histograms and heat maps. Also, I learned the phylogenetic tree analysis and the variant calling. So on the basis of this, the tentative topic I decided was, with the help of my mentor, Harpreet Kaur, is bioinformatics approach to analyze variants in retinitis pigmentosa. So what is retinitis pigmentosa? First of all, it is a heritable genetic rare genetic disorder. It can be inherited through three inheritance patterns, that is autosomal dominant, autosomal recessive, and X-linked. That is why I think it became my topic of interest. The occurrence probability is 1:4,000. And in this, firstly, the road receptors are affected and then the cone receptors. Approx 100 genes are mutated to cause this retinitis pigmentosa. Some are mentioned here. The sources are mentioned too. There are three stages mainly. Number one is the night blindness. Number second is there is a visual field loss of the concentric. Then it is the central vision loss and the person becomes completely blind. So I came across a fact actually. It is almost 60% of causal variants are unknown for RP till date. And the two questions which came across are how mutated or absent genes result in vision loss, and is the gene under study homozygous or heterozygous for the disease retinitis pigmentosa in given samples. I had gone through the methodology. It is like the samples are collected and they then the whole exome sequence is had been performed. So after that, each sample had gone through the variant calling. Then all the VCF files are combined, and then the joint calling had been performed. After that, the variant filtering is done, and using the plink, it had been distinguished that whether the gene is homozygous or the heterozygous. This is the presentation, and now I really want the suggestions that how to download the data sets or how to look for the data sets to run the variant callings and all. Thank you.
Thank you, Jeevan. Yes.
Okay. Okay, maybe I can jump in here. So if I understand correctly, you've identified your question, but you don't really have a data set that you found. Have you looked at any data sets at all?
Yes, sir. Sir, I just looked for a data set that is SRA of PRPF gene in retinitis pigmentosa.
Just one gene?
Yes, sir.
Okay, so what I would suggest, and again, not having the time to kind of think and see what's out there, but I would suggest that, first of all, one gene and the specific mutations you could just find on reference databases. You don't really have to do the analysis itself. So typically, we can help you find some data sets, but I would say that the more interesting question would be to first demonstrate that this gene is in fact the most important. And the second thing is to really try to understand what types of projects recently have been done to look into this. I mean, is this an unanswered question? Is this something that no one has ever asked before? Or has somebody asked this and they've come to some results, and in those results, maybe there's some contradiction or a gap, something that hasn't really been addressed? So I think that's kind of where you are. And so I would do, just like you did literature review, do a review of available data sets on this topic.
Okay, sir. Sir, the causal variants are not known till date. Like 100 genes are being mutated in this, and I just mentioned that 60% of the causal variants are unknown. So that's why I just chose the topic to study more about it.
But if they're unknown, how do you know it's in this one gene?
Okay, sir. So means I have to study for genes more than one.
Well, yeah. So I mean, if it's unknown, so you have a disease associated with some mutation, let's say, right? Or you suspect that it's a mutation. Now you're saying that I'm going to take this one gene and I'm going to look for everything in this gene that is, maybe associated with a disease. I think you have to step back and you have to ask yourself, so how does a phenotype how does the association between genotype and phenotype gets found in the first place? So maybe you can say that, many proteins are involved in function of a particular organ or a tissue. And here I have a disease, which is maybe some bad function that is being affected. So your question is why, right? And so what you need to have is, again, just like Dhruv presented, you have multiple conditions, maybe healthy and sick. And then you can start asking the question based on the comparison in groups that I have, what are the different factors? So maybe it's a mutation, maybe it's a methylation, maybe it's gene expression, maybe it's something else. So that's where I think you are. Now you took it to mutations only, and maybe that's because there's some genetic association that is known, right? That's what you were trying to say, that there's a known risk associated with genetic inheritance. So in that case, maybe you need to kind of think of a specific criteria. How do I find a data set that actually has those kinds of factors already in the data? Have you looked on NCBI for any data sets at all?
Yes, sir. So I found it one data set only till date.
What is it? Do you have a link?
So I could share the screen for that.
Mm-hmm.
So may I share the screen?
Yeah, sure. Okay, so this is a gene expression data set, right?
Yes, sir.
Actually,
Okay, so
it is not a gene expression data, Ilya. Because she showed me it was not gene expression data, as much as I remember.
It says here expression profiling by high throughput sequencing, but okay.
Can you just scroll down?
And here's a suggestion, also a useful suggestion from Iman. She's saying that you could potentially look for also animal model data, so like mice. She says that she found some geo data sets for humans and for mice, but I guess we are kind of going back to that original question. Gene expression data will give you a sense of which genes are involved, and sometimes a gene with a mutation is expressed, but it's not performing its function correctly. So that could be one cause of disease. Sometimes it could be that the mutation itself, let's say a stop codon, changes the full protein. So the question is, what are we really looking for and why? And if you focus on VCF files for DNA sequencing data, maybe that's a good place to start because you know that there's genetic inheritance. And if you do that, there is a analysis called genome-wide association. If you have enough data, and here you need to have maybe at least hundreds of data points, so whole genomes. So you can look up GWAS, and if you have maybe hundreds of samples with genomic data, and you can quantify the phenotype, here you need to think about what does it mean to quantify the phenotype. For example, GWAS is used oftentimes in resistance to different drugs. So there you have each drug and resistance, full resistance, partial resistance, no resistance, right? So now you need to think, how can you assign numbers? So if you have a set of genes and mutations, and maybe it's multiple mutations together, and you have the phenotypes, the model of GWAS finds associations that are statistically significant. I think that if this is still unexplored, there is not a known mutation for the cause, maybe that is because there's not enough data, or maybe because this phenotype is difficult to quantify. So maybe that's another challenge. But I think that we do have a project on tuberculosis that describes GWAS in detail. So if you go and learn and you just want to understand how GWAS works and how to deal with mutations, I would suggest we have a course on genomics that talks about cancer, right? And I saw that you went through that already.
Yes, sir. Yes, sir.
Yeah. And then, so GWAS is a technique that, again, is very specific to the type of data that you have. You have to have a lot of samples and you have to have quantified phenotypes. And Harpreet actually has worked on the tuberculosis data set, so I think she can explain that and see whether that's applicable in your project.
Okay, thank you, sir.
Harpreet, I don't know, do you want to mention something about that?
Yeah, I think I just need to, yes, I will discuss it with her. Because I need to understand exactly what questions she wants to ask. So that was my, maybe I'm not able to actually got her exact idea what she wants to work on. Because she asked that she wants to identify variants. So that is why I just want to make sure that we should able to understand exactly what the questions she wants to work on, because the data set, I think still we can help her to find out, but only I'm just not able to understand exactly what she's trying to find out in her project.
But I think definitely it's a good start, right? So it looks like you've done some background analysis, you kind of identified that this is an interesting topic for you to work on. Now we need to, I think, quickly move on and focus on the type of data. What data set could you work with?
Yeah.
Yes, sir. So it's a request as we are very new to this. Like I had not gone through any research before, so it is like I'm not asking for the spoon feeding actually, but it's like we are very new. Like we know we want to know a lot of things in this. Like my peers also in batch 2.
Yeah. So I think that's why it's very useful to take an example first and go through that example. And then you understand how people have gone from a general topic of interest to maybe asking that specific question when they have the data already available. So I think that what we are trying to do here is, before you jump to asking the question about this new topic that you've discovered, know how to ask the question. And then once you know how to ask the question, then you know that before you even can ask about a gene or a mutation, you need to show us that the gene or the mutation is the way that we should be addressing this question that you have in mind. And in my understanding right now, you have gotten to a place where maybe with your peers, maybe with others that are interested in this question, and maybe you can convince them that this is a useful question to pursue, then you can ask them, let's think together, let's brainstorm, let's look on NCBI, let's find other data sets and publications, and let's come up with an interesting question to focus on, because all of you will spend a lot of time, right, trying to work through the difficulties and finding some answer. You want to use that time in a meaningful way.
Yes, sir. So do I have to look the transcriptomics portion also for this, or it's just the genomics one?
So I don't know anything about this disease myself, so I can look into it myself also, but I would like to know why genomics or why transcriptomics before I say, go for transcriptomics, right? So, I mean, from the data set that you showed, it looked like it was a response to treatment. So when you treat some cells, you can measure gene expression. But genomic data maybe is more relevant to the cause. Maybe it's a genetic inheritance. So those are two very different questions, and that's why those two different data sets would be very different. If you want to understand the cause, you probably want to look at which genes are significant, and then you want to go back and say, where are those mutations? Cause is a very difficult question in biology because we don't have all the information to study the cause, right? We only have some associations that we can find, and those associations are not always the cause. There might be the effect of something else that we don't see. And so the cause and the mechanism are some of the major challenges in biology, but the effect and the application are sometimes easier, because you can say, I have a treatment, I have an effect, I can measure both of them. And then if I have an association, maybe I can use that in a meaningful way. Or if you want to learn about something that has to do with the impact of something already known, like I know there's a mutation, now I want to understand exactly how that works in a specific context, right? Those are questions that are a little bit more easy to address with the data analysis itself. But in your case, I wouldn't say that that's to say that you shouldn't pursue the question you've identified. It sounds like an interesting topic. It's new to me. I'm sure it's new to a lot of other people. Let's find out what's in there. I think that's the purpose here, is to be able to ask these questions.
Thank you, sir.
Sorry.
Thank you so much, Ilya, and thank you so much, even Jyota, as well. So now I think we should be ending this meeting. We had a great discussion. We discussed about our research fellowship program, and also we had our research fellows present their work. Though we have a lot of research fellows to present, but just time is the limit, and that's why we just had a few of them. And also we have a lot of new participants today. We have a lot of new people who want to be a part of the research fellowship program. So I'm sharing with you all the registration link of the program, and you can register yourself here for the program. And if you have any doubt, then you can mail me at marketing@pine.bio. Iman, we'll just take your question. Yes. Okay, so your question is, if the timing does not fit your schedule, then what, right? So yeah, it doesn't matter. So Iman, you can anytime schedule a one-on-one meeting with Dr. Harpreet and then discuss your research work. That would be better for you. And these research fellowship meetings, we basically have to discuss to have the presentations by a research fellow, so that also you all get to know what's happening and what could be done further, and also you can clear your doubts here. But you can anytime ask your mentor to schedule a one-on-one meeting and meet at your time, at your own time. You can have a word with Dr. Harpreet at support@pine.bio and let her know what day and time suits you, and then she'll fix a meeting accordingly. Yes, it would be according to your schedule, Iman. I hope that sorts your doubt, right? Okay, thank you. And for others, you can register yourself on the link I've shared, and also to learn more about the program, and also to have insights from the work of our previous research fellows. You can visit this research fellowship blog here. It has all the details, what all would be covered, what would be the process of the research fellowship program, and also it has the sample projects and the projects from our research fellows, our ex-research fellows, and seeing them all, you can have great insights and learn more. And also, Ilya, we have people here from LBRN group today. So Rizwana, I would want you to introduce yourself to us.
Hello.
Hi, Rizwana. Yes, you're audible.
Yes. Hello, everyone. I'm Rizwana. Yeah.
Thank you.
Hello, everyone.
Yes.
I'm Rizwana. Currently, I'm pursuing my PhD degree in environmental toxicology at Southern University and A&M College, Baton Rouge. And my dissertation project basically focused on vaping-induced toxicity. And we basically focuses on molecular mechanisms. And in that, I am particularly targeting proteasome and immunoproteasomes. These are the two cellular pathways which are responsible to maintain proteostasis. And in this specifically, I'll be determining the assembly of mixed proteasome variants with constitutive and inducible subunits. And I'm also interested to include the RNA sequencing and the chip sequencing analysis in my thesis. So that's the reason I have joined this research fellowship program.
Okay, and Rizwana, have you created an account today? On Learn, did you create an account?
Yes.
Okay, great. So if you could paste your account link, the URL from the top URL, just paste it in here so that we can add you. We need to submit it to LBRN.
Okay.
Thank you.
Thank you, Rizwana. Uchechi, can you please introduce yourself next? So I think Uchechi can't do. So Shilpa, can you introduce yourself, please?
Hello,
Hi,
introducing
Shilpa.
myself now. Oh, it's Uchichi.
It's Uchichi. Oh, I'm so sorry. It's Uchichi.
Oh, it's okay. It's okay.
Yes.
I'm having internet issues. That's why I couldn't hear.
Oh, it's perfectly fine. Yes, can you please introduce yourself?
Yes, ma'am. Good afternoon. My name is Uchechi Owuna, and I'm a senior toxicology major at the University of Louisiana Monroe. I'm currently working with Dr. Moreau in his lab where he's working with 1,3-diarrhyl and we are synthesizing the pyrazoles and conducting biomedical research on them for the treatment of melanoma skin cancer. So my research interest is I'm trying to research the field of psychopharmacology because I've taken a lot of pre-pharmacy classes and that's something I've always been interested in. I've been interested in seeing how the post and antenatal medications affect postpartum depression. And I'm really hoping that with the skills and techniques and knowledge I learned from this program, I'll be able to apply it into this research. I'm also hoping that I'll be able to use this research to learn how to develop this project and also use this experience to make connection and learn more about postpartum depression and how to properly research topics that cross between psychology and pure science.
And also, could I ask you, did you make an account today?
Yes, I did.
Okay, perfect. So if you can post the link, the URL link to your account so we can report that. Thank you. Now, did I hear correctly? You were interested in melanoma cancer, but also depression?
Oh, I said I'm currently working on synthesizing 1,3-diarrhyl for treating melanoma skin cancer, and that's the research work I'm doing now. But in the future, I'm trying to build this other research idea that I'm very interested in, but I haven't really been able to find material and support for that. So that's another reason why I decided to join this course and the program.
Okay, perfect. So one thing I wanted to mention to everybody who is joining now, every week we ask that you submit a progress form. So we have this research fellowship weekly progress report. This is an important part of what you will be sharing with us in terms of your progress. So I'm just going to share the link in the chat and we will follow up with an email so that you can take a look. This form allows us to understand where you are in your progress and helps you structure your progress and ask for meetings one-on-one that Sonalika mentioned already. Yes, so thank you, Sonalika, for sharing that link too. And in here, you will see different stages. Those stages are kind of where we can help you by identifying the type of help that we can direct you to, either a free resource or something that we've designed specifically for that, or how to schedule a meeting, like I mentioned, with a mentor. Okay, thank you.
Thank you, Uchechi. Now I would want Shilpa to please introduce herself.
Hi, everyone. Hello, everyone. I'm Shilpa Tota. I was born and brought up in India. I did my master's in the field of biochemistry from Kakya University, India. Presently, I'm pursuing my PhD in environmental toxicology department at Southern University and A&M College, Baton Rouge. My dissertation work mainly focuses to explore the effect of pulmonary and hepatic toxicants like pentachlorophenol and heat shock proteins and autophagy process in humans. In terms of molecular mechanism, I'm targeting mainly on protein homeostasis mechanism and on epigenetics too. I want to include my RNA sequencing in my dissertation work. So that's why I joined this class to learn about the data analysis and sequencing. Thank you.
Okay, sorry, we could not actually see your video. Maybe it's dark or maybe your camera is not working, but either way, it's fine. So again, were you able to complete your registration today?
Yeah, I did.
Okay, perfect. All right, great, thank you.
Thank you.
Thank you so much, everyone, for joining the meeting today. And we would be meeting soon. I've shared the links for those who want to join the program. And if you have any sorts of queries, you can mail me at marketing@pine.bio. I'll just type it in again for you. And I'm looking forward to hearing from you. Thank you so much for joining.